let me begin first with the biological context in which this problem was formulated and we did the calculations. So we have about 10 to the power 15 chemical synapses in our brain, and each one of those synaptic junction looks more or less schematically like this. You have some neuron coming all the way from the left and terminating here, and here is another neuron which starts and goes all the way to the right, let us say. And how does a signal transmit across this gap? So usually what happens is that if you have a sensory stimulus, whether it be auditory, visual or mechanical or even directly electrical given in an in vitro experiment, that will cause a depolarization of the membrane potential which propagates. It's called an action potential. So it comes all the way and it reaches this terminus, and then something happens, which is that some channels open up and some calcium ions come in, which trigger these bags, these balls that I show schematically, which are called synaptic vesicles, to be fused and released in this space. So what do they release? These black dots, which are called neurotransmitters, and those are taken up by receptors here and made into another electrical pulse which converts and goes. So all these you don't have to remember for the rest of the talk. Let us focus only on this part, that some bags were docked here. Those some of them got released in this space, and I would be interested in the statistics of that. So the fact that such transmission involves release of certain number of quanta of these molecules, neurotransmitters, was first realized around 1954, and within 20 years experimentally there was a consensus that the statistics of single release is a binomial. So let me explain what is being told here. If suppose there are n- number of docked vesicles at any given instant of time, then the probability that B of them are released is given by this. So PR is the release probability. Okay, so now we would be interested in a more complicated situation in which there is not a single release but a sequence of such releases. That is because very typically in experiments, for example, they may use a frequency to give these spikes. So you will have these action potentials coming at equal intervals, or in general physiological conditions there may be random intervals between these action potentials at intervals of Ts, which may be random, and those spikes would be coming. Each spike will lead to a release of certain number of vesicles whose height is shown here. So this quantal content is nothing but this number B, okay, remember. And so we are seeing certain parameters that are already emerging in the model. One is a release probability. Another is, of course, in between two spikes there will be more docking of the vesicles. So there would be what we'll call as the replenishment of the vesicles on the membrane. There would be some rate associated with that K. And of course there will be certain maximum number of docking sites here, which I have not written here as a symbol, but capital M. So please remember these three quantities PR, K and M, which I will be repeatedly talking about. Moreover, if you look at a typical signal like this, I will show you actual signal from experiments in a few slides, but they will look somewhat like this. There will be some large spikes which will reduce then to smaller spikes and become steady. So there is a transient region and there is a steady state. Why steady? That is very easy to understand, that at some stage the release and the replenishment somehow would balance out on an average. So there has to be some kind of steady state. In general, one would expect that. So the question is that what is the statistics of this released number of vesicles? Whether it be in the transient state or the steady state, can something be told about it? So we looked in the literature, and in the next 50 years, quite surprisingly, we did not find any clean treatment of this problem. And I will tell you reasons for that. But we came across lots of ad hoc answers. People keep using the binomial for this whole statistics, replacing this n- with an average n-. Sometimes they use that capital M. Sometimes they say that when the stimulation is not done at equal intervals, it's not a binomial, they do some approximation on it, but there isn't a clean treatment that we found. So we thought here is a problem, one can try to look at it. Is it a binomial or not from our mathematical perspective? But there is a very important neuroscientific interest in this. You see, if you can find the right statistics, then the moments or distribution, they will depend on these parameters, and these parameters actually characterize a type of neuron. So if the neuron is diseased, for example, these parameters will change. So you can actually measure the moments experimentally, compare them, drag out these constants, and then comment whether the neuron is healthy or not healthy. Okay, so here is the mathematical version of the problem. So if the number of docked vesicles is, let's say, nt as a function of time, after one of the action potential, typically it will be low, then before the next action potential it will rise, then there will be a drop because of the release, then there will be again a rise, again a drop, and again a rise. So this will continue repeatedly over different cycles, where these are the time intervals between two action potentials, m-1th and the mth one. So how one can proceed is that one can say there is a probability, for example, of this number n plus. So notations first. N plus is the number nt just after an action potential. N minus denotes just before the action potential, and bm is the difference. Okay. So, but as you see, this number n plus depends on the cycle m, n minus b all depends on the cycle m. Similarly, for the previous cycle, there are other numbers. So one can start with the probability of this number n plus here, and then there will be a propagation of that number stochastically up to the number here, which is given by this, and then there will be a binomial release. And then if you sum over this number and this b, then you should get the probability of this number n plus. Likewise, you can get the probability of this bm if you know the probability of this n minus, for example, multiplying by a binomial and summing over n minus. If you want the probability of the n minus, that's the first line that you can get from building up from the n plus here, multiplying with the propagation of that number from here to here. So all these are fine. So then why is the problem? It is not that we wrote these equations first. We saw that in the literature, similar equations were written down, little bit making it more complicated because they have to be more faithful to the biology, but in essence these equations were there. So what is it that stopped them from solving? So one of the things that they point out is that for this to solve, actually because there is the history dependence, m related to m-1 to m-2 and all that, one should at some point know the probability very correctly for some initial time. And that is where they sort of say that until and unless a neuron has been resting for very long, how do you know for sure that this is the probability? Even probabilistically this number is not known. So that is a reason why people have ignored really solving it. But then our answer to this problem was that why don't we look at the steady state, where I don't have to look at the memory. At least there can I give an answer. And then models, of course, one has to define this growth process, whether it is a simple process or what process. Then parameters will be involved. Can those parameters be estimated? Since they did not calculate in the literature, people were using Bayesian methods to estimate the parameters. And of course, then there is the question that these times can be correlated. So this is just to show you that in this project we had experimental data of this particular synapse from juvenile mice's brain, and this blue trace is the trace of this quantal content B. What you see here, this blue are 100 points over a second, and the bursts are coming after every 10 milliseconds because the train that was used was a 100 Hz train. So and the red line is an average over many such blue curves, and the point of this slide is that yes, there is a transient period, but then there is indeed a steady state. And so let us focus on the steady state and try and see if we can give an answer to this problem. So in order to proceed, we had this equation. So now you can use the statistics of these time intervals, these Tm's and Tm-1. So one can multiply with the joint distribution of Tm and Tm-1, G2, and then integrate over these times. So you can get on the left hand side the steady state probability of the number just after a release. On the right hand side, I cannot pull it through until and unless I demand a product, breaking of this 2-point function into two 1-point functions. This is the uncorrelatedness assumption that we use to proceed further. Okay, so if you proceed, if you make that, then you have the self-consistent equation between the probability of the number just after an action potential being related to itself, the same type of function on the two sides, so at self-consistent equation. E minus, which is the number, the probability of the number just before an action potential can be related to just after, and the burst can be related to P minus. So basically there are these functions Q, P minus and P plus that we have to calculate, and they are all related to each other. The main equation is here, the self-consistent equation, which if we know the P plus, then the rest are known. Okay, so again I am just for ease writing down the same equations just to point out that what we are interested is that, is this really a binomial as people have been using in the literature? Maybe it is. After all these clumsy calculations, maybe it will be so. But later we have to check. This is a binomial known. Now, this GTS is the important distribution of the interspike spacings. That is, what is the distribution of the times after which the action potential comes. That has to be given to us by the experimentalist. So we'll keep that completely general. We have to converge on what is the model of building up the number. And there we looked and found the simplest model in the literature. So we did not go to any complicated model. Of course, for every complicated model, you have to do a full calculation. But the simplest model was as simple as this. You have maximum number of docking sites. If nt is the number of docking sites at any given time, m minus nt is the number of empty sites. So you can just say that the rate at which n goes from n to n plus 1 would be k into m minus n. So as simple as that. So thus you write the master equation, its solution of course you know. Then we can proceed further, that we can build generating functions for these p minus to be f minus, qb to be fb, and f plus for p plus. And as I said that there would be a self-consistent equation now for the f plus. Once you know f plus, you can build the generating function of this fb, and from there you can get the desired distribution. So now what is happening here, this q is getting mapped to another point q prime. This is a homography map, and but its fixed point is q is equal to 1. So we thought it good to expand the two sides in Taylor series with coefficients this f plus n, which we now have to figure out, but a series about q is equal to 1. So then recursively we figured out that these coefficients have this form. It's a little bit clumsy looking form, but let me explain. I mean, there are few functions here. So q is this phi function and the L function. The L function is nothing but a Laplace transform of GTS, that is the times time gap distributions, and phi mn is this integral over GTS. And you can once you know GTS, you can calculate these objects. What is the sum like? Sum has terms. Each one of those terms involve a set of integers mz minus 1 up to m1, and those integers are nothing but subsets of the number from 1 to n minus 1. Okay, so this is what we have. It's a clumsy formula, but once you know the f plus n, you can go back and from here get the f plus q. From the f plus q you can get the fb, then you can take the derivative with respect to q and set q is equal to 0 and get qb. Okay, so that's the procedure. One can do that, and finally this is the qb. This is the formula for any interspike interval GTS. We get this, and generally it doesn't look to be so simple that it will reduce to a binomial. One has to check, but it's a clumsy looking formula. Usefulness is that once you know the full distribution, you can get all the moments. So here are the first two moments, and as you can understand that if experimentally somebody knows the mean, let's say the CV square or the variance and the skewness and all, then these parameters m, pr and k is hidden within these L's and the phi's, those can be figured out. So now we will move to special cases. This is a very popular case, that is the spikes come at equal intervals. You take a Dirac delta. When that is so, these functions can be evaluated exactly. This series by mathematical induction can be reduced to this, And when we put all that, to our surprise, we got a binomial. Okay, so for equal interval times, it is indeed a binomial, but the release probability is a modified release probability. It is no longer PR, but it is this complicated function which now depends on the replenishment rate K and capital T. So for years people have been using binomial, fitting to binomial, but this is the right binomial one needs to fit to. And if one uses any ad hoc binomial like this, I mean, you can see this is the exact formula and the Monte Carlo. This green curve is this ad hoc binomial. It will not match with the exact result. Now, for this constant time interval data, one problem is that this parameter estimation, you may get stuck because the parameters of this probability distribution is capital M and PRB, which actually lumps up all these constant K and PR together. So you don't have independent way of determining K and PR. That's a problem. But let's say whatever we could do, I will tell you. This is the, from lots of data for this particular synapse. We had 400 sample data points from experiments. This is the green histogram from experiments. This is the curve theoretically. To compare theory and experiments, of course, you have to match something. So we matched the mean, and in a particular way, we estimated this M, not from the steady state data, but from the first release data, because we were told by the experimental collaborators that this between every data, they gave the neuron a lot of rest. So if you give it a lot of rest, then you know the first release should be according to this binomial given by this capital M. So M can be obtained from there. And then using those two parameters, we matched the Kalbeck-Leibler divergence. It is small. Then of course, since you knew that this functional form is like this, dependent on the frequency, we looked at the data published in the literature for 3 different synapses. We saw these data points were available. We put our theoretical curves. They seem to be matching well. And then we sort of moved to more more physiologically important cases, because naturally speaking, the spikes should not come at equal intervals. There will be some randomness. So one of the cases that we could handle was these exponentially distributed gaps. Then these functions. Then also this sum that we had, luckily we could by induction reduce it to this, and putting it together, the distribution looked much simpler, and it is not a binomial. So this is one of the central results that indeed it is not fair to assume that it's a binomial distribution at all. And this is the exact distribution, and these are the kinetic Monte Carlo. And if one thinks that I will force a binomial on it with by assuming this 1 by f naught to be an average time, then that would be the dashed curve, which will go nowhere near this actual curve. Also for this exponential distribution, if the moments and that you have, they have this K and PR and M all separately appearing in the formulas. So that is very good. Actually, if we had experimental data for these mean variance and so on, then by comparison one can actually get the values of these parameters, which is the more important thing in this neuroscientific context. So that's that. I mean, one can look at gamma distribution also, so it had these. But now coming to more practical concerns. So what about correlations? So this, of course, we could not solve exactly. What we did was we simulated 2 models in which we tried to produce correlated data. So the mth time was chosen from a random exponential, from a random number from the exponential distribution as a fraction, and another fraction we added just the time before. So the mth time depended on the m-1th time, and thus it got correlated. Similarly, the second model you correlate with times which are from all the way from 1 to m-1. Different kinds of correlated models can be created, and this epsilon gives you a handle of how to tune and make the correlations higher. So the blue is the uncorrelated exact result that we have, but then as we tuned up the epsilon from 0.1 to 0.8, For point 1, it doesn't look that different, but as you go to point 8, these green and the orange curves, they start definitely deviating as expected from the uncorrelated result. Of course, if one cares about relating to biology, then there are more complicated things one has to address. Already in the past 10-15 years, it has been realized that this release I was talking of a single kind of synaptic vesicles is not true. Typically, there are multiple pools. Then there are complicated processes that the vesicles after released comes back and refuses. So there are feedback processes. Then what I am calling as the quantal release gets converted into a current. In between that, there is stochasticity, so that also can be put in. But our point was that all these complicated things can be done once you have done something simple, and that is what we were trying to get to. So this paper has been published here. This was my PhD student. He has gone for a postdoc now, Krishna. And these were the experimental collaborators who gave us a little data. They did not want to do fresh experiments on the Poisson case, otherwise we could have compared. And these two persons helped me to understand the neuroscientific literature a lot of it. So in conclusion, for the simplest model of synaptic transmission on sustained stimulation by random uncorrelated action potentials in the steady state, so only in such regime we have been able to give an exact distribution for this quantal content, and typically it is not a binomial. For the fixed frequency case, it is indeed a binomial, but the effective release probability is a complicated function of all of these. If it is an exponentially distributed ISI, we have explicitly shown it is non-binomial, and thus it would be nice to compare at least this case to experiments on electrophysiology of synaptic.
Dibyendu, very nice talk. So this, in the case of this delta function spikes, right? So you get this release rates, right? This PR. So it becomes an effective function of the rate at which the docks were filled up, right?
Yes, and also a function of the frequency at which you are stimulating.
Right, the frequency, that's what I mean. But it doesn't depend on the number, like the occupancy of this, how many, these replenishments, whatever these guys are. Doesn't it depend on the number of...
The fluctuating number has been integrated over. So if I may go back to answer
this PR, essentially, it doesn't depend on this n minus, is it?
No, the whole game was to integrate over the n minus, right? See, that's the stochastic process we have been integrating. You see, finally you will be left with the parameters of the model, no? N minus was a fluctuating quantity.
Right. So I thought the probability also will depend on n minus. That is somehow I thought. I mean, initially when you started building up the model, not only the rate at which the replenishments are done, but also like the number of occupancies.
Maybe it was
like the first, first couple of slides, maybe.
I go here. Yes. Yeah. But they have been summed over, right? So they have been summed over. Those are stochastic numbers which have to be summed over. At the end of the day, I will be left with the parameters of the model. So
thanks.
May I? Okay, so you said you can distinguish between healthy and unhealthy neurons based on this study.
Yes.
But you have 3 parameters. So which one is not working properly, or can you
first of all, we have to kind of standardize. Like if there is a data with this Poisson train that we have, first we have to check if the distribution we are predicting matches that, because there are biological neurons will be more complicated. If at least our data matches, like this fixed frequency matched quite well, but now if the other one matches, so that will give us some idea that things are working fine, even this reduced model is good. But next one has to see if that same type of neuron is diseased, then the numbers that will come out from the same statistical formulas will be different. So it would be one way to check that, if somebody finds that parameters are pretty different, but when they are healthy, they will have a particular set of numbers, right? Okay. Which are characteristic of the neuron. For the diseased one, it will be different. That's the point I'm trying to make.
Long live India and France friendship.